All right, so welcome to the first video of Biology 169, Anatomy and Physiology 2. In this one, we're going to be talking about Module 1.1.1. This is basically the first module in the endocrine system. We'll split the endocrine unit into two units. This will cover the first part of the first week. There's 3 modules inside of this particular one. In this series, we'll look at the endocrine system, intercellular communications, and hormonal actions. So the human body contains about 30 chemical messengers that are known as hormones, and these regulate activities such as sleep, body temperature, hunger, stress management, all sorts of things. These hormones are a product of the endocrine system, which, along with the nervous system, controls and coordinates our body's processes. As we get further into this particular unit, we'll examine the structure and functional organization of the endocrine system and compare it to the nervous system. After an overview of the endocrine system and the characteristics of the hormones that it produces, we'll consider the structure and the function of the body's various endocrine glands. And then finally, we'll look at some of the ways in which the hormones modify metabolic operations and the interactions between the endocrine system and other body systems. So let's begin by kind of considering the role of intercellular communication and maintaining homeostasis, which is one of our main concerns here. In these couple figures, you can kind of see where and how the endocrine system is laid out. All right, so a key point to remember is that homeostasis is preserved through intercellular communication. In this table, table 18.1, it summarizes the 4 ways that cells and tissues communicate with one another. Viewed from a general perspective, the differences between the nervous system and the endocrine system seem relatively clear. In fact, these broad organizational and functional distinctions are the basis for treating them as 2 separate systems. Yet when we consider them in detail, we see that the 2 systems are similarly organized. Both systems rely on the release of chemicals that bind to specific receptors on their target cells, and the 2 systems share many chemical messengers. For example, norepinephrine and epinephrine are called hormones when released into the bloodstream, but called neurotransmitters when released across synapse. Both systems are regulated mainly by negative feedback control mechanisms, and the 2 systems share a common goal, which is to preserve homeostasis by coordinating and regulating the activities of cells, tissues, organs, and systems. To preserve homeostasis, cellular activities must be coordinated throughout the body. Neurons monitor or control specific cells or groups of cells. Only a small fraction of all the cells in the body are innervated, however, and the commands from the nervous system are often very specific and relatively short-lived. Yet, many of the life processes are not short-lived. For example, your body takes decades to reach adult height. The body continually controls and maintains its reproductive capabilities for at least 30 years in a typical female, and even longer than that in males. Long-term processes such as growth, development, or reproduction involve or affect metabolic activities in virtually every cell and tissue. There's no way that the nervous system can regulate such processes. Instead, the endocrine system provides this type of regulation. It uses chemical messengers to relay information and instructions between cells. To understand how these messages are generated and interpreted, let's take a closer look at how cells communicate with one another. In a few specialized cases, adjacent cells coordinate cellular activities by exchanging ions and molecules across gap junctions. This direct communication occurs between 2 cells of the same type, and the cells must be in extensive physical contact. The 2 cells communicate so closely that they function as a single entity. Gap junctions have several functions. 1. Coordinate ciliary movements among epithelial cells. 2. Coordinate the contractions of cardiac muscle cells. And 3. Facilitate the propagation of action potentials from one neuron to the next at electrical synapses. Most communication between cells involves the release and receipt of chemical messages. Each cell continuously talks to its neighbors by releasing chemicals into the extracellular fluid. These chemicals tell cells what their neighbors are doing at any moment, and the result of this coordination of tissue function at the local level. This use of chemical messengers to transfer information from cell to cell within single tissue is called paracrine communication. The chemicals involved are called paracrine factors, also known as local hormones. Paracrine factors enter the bloodstream, but their concentrations are usually so low that distant cells and tissues are not affected. However, some paracrine factors, including several of the prostaglandins and related chemicals, have primary effects in their tissues of origin and secondary effects in their other tissues and organs. When these secondary effects occur, the paracrine factors are also acting as hormones, which are chemical messengers that are released into one tissue and transported into the bloodstream to alter the activities of specific cells in other tissues. Most cells release paracrine factors, but only specialized cells produce typical hormones. Nevertheless, the difference between the paracrine factors and the hormones is mostly a matter of degree. Paracrine factors can diffuse out of their tissues of origin and have widespread effects, and hormones can affect their tissues of origin as well as distant cells. By convention, a substance with effects outside of its tissue of origin is called a hormone if its chemical structure is known, and a factor if that structure remains to be determined. In intercellular communication, hormones are like messengers, and the cardiovascular system is an email. A hormone is released into the bloodstream, it is distributed throughout the body. Each hormone has a target cell, specific cells that the receptors need to bind to and read the hormonal message when it arrives. But hormones are really like email spam. Cells throughout the body are exposed to them whether they have the necessary receptors or not. At any moment, each individual cell can respond to only a few of the hormones present. The cell ignores the other hormones because it lacks the receptors to read the messages they contain. The activity of hormones in coordinating cellular activities in tissues in distant portions of the body is called endocrine communication. So, how do these hormones work? They alter the operations of the target cell by changing the types, the quantities, or activities of important enzymes and structural proteins. So, a hormone may stimulate the synthesis of an enzyme or structural protein not already present in the cytoplasm by activating appropriate genes in the cell nucleus. It can increase or decrease the rate of synthesis of a particular enzyme or other proteins by changing the rate of transcription or translation. Or it can turn an existing enzyme or membrane channel on or off by changing its shape or structure, which is called a conformational change. Through one or more of these mechanisms, a hormone can modify the physical structure or biochemical properties of its target cell. Because the target cell can be anywhere in the body, a single hormone can alter the metabolic activities of multiple tissues and organs at the same time. These effects may be slow to appear, but they typically last for days. Consequently, hormones are effective in coordinating cell, tissue, and organ activities on a sustained long-term basis. For example, circulating hormones keep body water content and levels of electrolytes and organic nutrients within normal limits 24 hours a day throughout our entire lives. And cells can respond to several different hormones simultaneously. Gradual changes in the quantities and identities of circulating hormones can produce complex changes in the body's physical structure and physiological capabilities. Examples include the processes of embryonic and fetal development, growth, and puberty. Hormonal regulation is quite suitable for directing gradual, coordinated process, but it is totally unable to handle situations requiring split-second responses. That kind of crisis management is the job of the nervous system. The nervous system, too, relies primarily on chemical communication, but it does not send messages through the bloodstream. Instead, as we have seen, neurons release a neurotransmitter at a synapse very close to the target cell that have the appropriate receptors. To command the release of neurotransmitters rapidly, travels from one location to another in the form of action potentials propagated along axons. The nervous system thus acts like a telecommunications company with a cable network carrying high-speed messages to specific destinations throughout the body. The effects of neural stimulation are generally short-lived and they tend to be restrictive to specific target cells, primarily because the neurotransmitter is rapidly broken down and recycled. This synaptic communication is ideal for crisis management. If you're in danger of being hit by a speeding bus, the nervous system can coordinate and direct you to leap to safety. Once the crisis is over and the neural circuits quiet down, things soon return to normal. Once the crisis is over and the neural circuits quiet down, things soon return to normal. So the endocrine system includes all of the endocrine cells and tissues in the body that produce hormones or paracrine factors with effects beyond their tissues of origin. As we noted previously in 168, endocrine cells are glandular secretory cells that release their secretions in the extracellular fluid. This characteristic distinguishes them from exocrine cells, which secrete their products into epithelial surfaces, generally by the way of ducts. The chemicals released by endocrine cells may affect only nearby cells, as in the case of most paracrine factors, or they may affect cells throughout the body. The chemicals released by endocrine cells may affect only nearby cells, as in the case of most paracrine factors, or they may affect cells throughout the body. So we can divide hormones into three groups on the basis of their chemical structure. 1, amino acid derivatives. 2, peptide hormones. And 3, lipid derivatives. Amino acid derivatives, sometimes known as biogenic amines, are relatively small molecules that are structurally related to amino acids, which are the building blocks of proteins. These hormones are synthesized from the amino acids tyrosine and tryptophan. Those made from tyrosine include thyroid hormones, which are produced by the thyroid gland, and the components of epinephrine, norepinephrine, and dopamine, which are sometimes called catecholamines. The primary hormone made from tryptophan is melatonin, which is produced by the pineal gland. We can divide peptide hormones into two groups. One group consists of glycoproteins. The other group is made up of short polypeptides and small proteins. There are two classes of lipid derivatives. We have eicosanoids and steroid hormones. Eicosanoids are signaling molecules and include leukotrienes, prostaglandins, thromboxanes, and prostacyclins. Steroid hormones are lipids structurally similar to cholesterol. The individual hormones differ in the side chains attached to the basic ring structure, and over time, the liver gradually absorbs these steroids and converts them to a soluble form that can be excreted in bile or urine. So let's talk about the secretion and distribution of hormones. Hormones are typically released where capillaries are abundant, and the hormones quickly enter the bloodstream for distribution throughout the body. Within the blood, hormones may circulate freely or travel bound to specific carrier proteins. A freely circulating hormone remains functional for less than 1 hour, and sometimes for as little as 2 minutes. It is inactive when it diffuses out of the bloodstream and binds to receptors on target cells. It is absorbed and broken down by cells of the liver or the kidneys, or it's broken down by enzymes in the bloodstream or interstitial fluids. Thyroid hormones and steroid hormones remain in circulation much longer, because when these hormones enter the bloodstream, more than 99% of them become attached to special transport proteins. For each hormone, an equilibrium state exists between its free and bound forms. As the free hormones are removed and inactivated, bound hormones are released to replace them. At any given time, the bloodstream contains a substantial reserve, generally around several weeks' supply of bound hormones. That leads us to some of the mechanisms of hormone action. Hormones coordinate cell, tissue, and organ activities on a sustained basis. They circulate in the extracellular fluid and bind to specific receptors on plasma membranes or within target cells. Binding modifies cellular activities by altering membrane permeability, activating or inactivating key enzymes, or changing genetic activity. To affect the target cell, a hormone must first interact with an appropriate receptor. A hormone receptor, like a neurotransmitter receptor, is a protein molecule to which a particular molecule binds strongly. Each cell has receptors for several different hormones, but cells in different tissues have different combinations of receptors. This arrangement is one of the reasons hormones have different effects on different tissues. For every cell, the presence or absence of a specific receptor determines the cell's hormonal sensitivities. If a cell has a receptor that can bind a particular hormone, that cell responds to the hormone. If a cell lacks that receptor, the hormone has no effect on that cell. Hormone receptors are located either on the plasma membrane or inside of the cell. And next, we'll use a few examples to kind of consider the basic mechanisms that are involved in that. So, the receptors for catecholamines, which remember are epinephrine, norepinephrine, and dopamine, peptide hormones, and eicosanoids are in the plasma membranes of their target cells. Catecholamines and peptide hormones cannot penetrate a plasma membrane because they are not lipid soluble. Instead, these hormones bind to receptor proteins on the outer surface of the plasma membrane, also known as extracellular receptors. Eicosanoids are lipid soluble, and they will diffuse across the plasma membrane to reach receptor proteins on the inner surface of the membrane. These are also known as intracellular receptors. So, with that in mind, we can consider first and second messenger. Basically, communication between the hormone and the cell uses first and second messengers. So, a first messenger is a hormone that binds to a receptor on the plasma membrane surface. A secondary messenger, or a second messenger, is an intermediary molecule that appears due to a hormone receptor interaction. So, basically, the secondary receptor or messenger happens as a result of the first messenger. There are some important secondary messengers, such as cyclic AMP, also known as CAMP, which is a derivative of ATP. You have cyclic GMP or cGMP, which is a derivative of GTP, which is another high energy compound similar to ATP. And you have calcium ions, which we know from the action potential release of calcium ions helps to control some of that ATP or action potential propagation across the cell membrane. So, when a small number of hormone molecules bind to membrane receptors, thousands of secondary messengers may appear in a cell. This process, which is called amplification, magnifies the effect of a hormone on the target cell. Moreover, the arrival of a single hormone may promote the release of more than one type of messenger, or the production of a linked sequence of enzymatic reactions known as a receptor cascade. Through such mechanisms, the hormone can alter many aspects of cell function at the same time. The presence or absence of a hormone can also affect the nature and number of hormone receptors and proteins in the plasma membrane. Downregulation is a process in which the presence of a hormone triggers a decrease in the number of hormone receptors. In downregulation, cells become less sensitive to high levels of a particular hormone. In contrast, upregulation is a process in which the absence of a hormone triggers an increase in the number of hormone receptors. In upregulation, cells become more sensitive to low levels of a particular hormone. The link between the first messenger and secondary messenger generally involves a G protein, which is an enzymatic complex coupled to a membrane receptor. The name G protein refers to the fact that these proteins bind GTP. In the figure here, it shows how a G protein is activated when a hormone binds to its receptor at the membrane. We're going to break down this picture a little bit further as well. What happens next depends on the nature of the G protein and its effects on secondary messengers in the cytoplasm. Roughly 80% of prescription drugs target receptors coupled to G proteins. First, let's talk about G proteins and CAMP. So, if you look at the left side of the figure that's shown, that's basically the steps that involve releasing or increasing CAMP levels. The activated G protein activates the enzyme adenylate cyclase, and then adenylate cyclase converts ATP to the ring-shaped molecule cyclic AMP. Cyclic AMP then functions as a secondary messenger, typically by activating a kinase. And a kinase is an enzyme that attaches a high-energy phosphate group to another molecule in a process called phosphorylation. Generally, cyclic AMP activates kinases that phosphorylate proteins. The effect on the target cell depends on the nature of these proteins. The phosphorylation of plasma membranes, or proteins there, for example, can open ion channels. In the cytoplasm, many important enzymes can be activated only by phosphorylation. One important example is the enzyme that releases glucose from glycogen reserves in skeletal muscles in the liver. Many hormones, including calcitonin, parathyroid hormone, ADH, ACTH, epinephrine, FSH, LH, TSH, and glucagon, produce their effects by this mechanism. And we're going to look at and talk about a lot of these hormones later on in this unit. The increase in CAMP levels is usually short-lived because the cytoplasm contains another enzyme, phosphodiesterase, which inactivates cyclic AMP by converting it to AMP, or adenosine monophosphate. The figure here in the center depicts one way the activation of a G protein can lower the concentration of CAMP within the cell. In this case, the activated G protein stimulates PDE, or remember, phosphodiesterase, activity and inhibits adenylate cyclase activity. Levels of CAMP then decline because CAMP breakdown accelerates while CAMP synthesis is prevented. The decline has an inhibitory effect on the cell because without phosphorylation, key enzymes remain inactive. This mechanism is responsible for the inhibitory effects that follow when epinephrine and norepinephrine stimulate A2 androgenic receptors. An activated G protein can trigger either the opening of a calcium ion channel in the plasma membrane or the release of calcium ions from intracellular compartments. The figure here in the right panel diagrams the steps involved. The G protein first activates the enzyme phospholipase C. This enzyme triggers a receptor cascade that begins with the production of diacylglycerol and inositol triphosphate from membrane phospholipids. The cascade then proceeds as follows. IP3 diffuses into the cytoplasm and triggers the release of calcium from the intracellular reserves, such as those in the smooth endoplasmic reticulum of many cells. The combination of DAG and intracellular calcium ions activates another membrane protein, protein kinase C, or PKC. The activation of PKC leads to the phosphorylation of calcium channel proteins, a process that opens the channels and permits extracellular calcium ions to enter the cell. This sets up a positive feedback loop that rapidly elevates intracellular calcium ion concentrations. The calcium ions themselves serve as messengers, generally in combination with an intercellular protein called calmodulin. Once it is bound calcium ion, calmodulin can activate specific cytoplasmic enzymes. This chain of events is responsible for the stimulatory effects that follow when epinephrine or norepinephrine activate A1 receptors. Calmodulin activation is also involved in the responses to oxytocin and several regulatory hormones secreted by the hypothalamus. Steroid hormones diffuse across lipid parts of the plasma membrane and bind to receptors in the cytoplasm of the nucleus. The hormone receptor complexes then activate or deactivate specific genes. By this mechanism, steroid hormones can alter the rate of DNA transcription in the nucleus. In this way, they change the pattern of protein synthesis. Alteration in the synthesis of enzymes or structural protein directly affect both the metabolic activity and the structure of the target cell. For example, the sex hormone testosterone stimulates the production of enzymes and structural proteins in skeletal muscle fibers and causes muscle size and strength to increase. Thyroid hormones cross the plasma membrane primarily by a transport mechanism. Once in the cytoplasm, these hormones bind to receptors within the nucleus on a mitochondria. The hormone receptor complexes in the nucleus activate specific genes or change the rate of transcription. The change in transcription rate affects the metabolic activities of the cell by increasing or decreasing the concentration of specific enzymes. Thyroid hormones bound to mitochondria increase the mitochondrial rates of ATP production. As noted earlier, the functional organization of the nervous system parallels that of the endocrine system in many ways. In 168, we considered the basic operation of neural reflex arcs, the simplest organizational unit in the nervous system. The most direct arrangement was a monosynaptic reflex, such as the stretch reflex. Polysynaptic reflexes provide more complex and variable responses to stimuli. Higher centers, which integrate multiple inputs, can facilitate or inhibit these reflexes as needed. Endocrine reflexes are the functional counterparts of neural reflexes. Endocrine reflexes can be triggered by humoral stimuli, which is changes in the composition of the extracellular fluid, hormonal stimuli, the arrival or removal of specific hormones, or neural stimuli, the arrival of neurotransmitters at neuroglandular junctions. In most cases, negative feedback controls endocrine reflexes. A stimulus triggers the production of a hormone, and the direct or indirect effects of the hormone reduce the intensity of the stimulus. A simple endocrine reflex involves only one hormone. The endocrine cell involved responds directly to the change in the composition of the extracellular fluid. The secreted hormone adjusts the activities of target cells and restores homeostasis. Simple endocrine reflexes control hormonal secretion by the heart, pancreas, parathyroid glands, and digestive tract. More complex endocrine reflexes involve one or more intermediary steps and two or more hormones. We will discuss this further in the next module, but just to tie things together, the hypothalamus provides the highest level of endocrine control. It integrates the activities of the nervous and endocrine systems in three ways. One, the hypothalamus itself acts as an endocrine organ. Hypothalamic neurons synthesize hormones and transport them along axons to the posterior lobe of the pituitary gland, where they are released into circulation. You should have discussed two of these hormones, antidiuretic hormone, ADH, and oxytocin, or OXT in your previous class. 2, the hypothalamus secretes regulatory hormones, which are special hormones that control endocrine cells in the pituitary gland. The hypothalamic regulatory hormones control the secretory activities of endocrine cells in the anterior lobe of the pituitary gland. In turn, the hormones from the anterior lobe control the activities of endocrine cells in the thyroid, adrenal cortex, and the reproductive organs. 3, the hypothalamus contains autonomic centers that exert direct neural control over the endocrine cells of the adrenal medullae. When the sympathetic division is activated, the adrenal medullae release the hormones epinephrine, norepinephrine into the bloodstream. The hypothalamus secretes regulatory hormones and ADH in response to changes in the composition of the circulating blood. The secretion of oxytocin, epinephrine, and norepinephrine involves both neural and hormonal mechanisms. For example, the adrenal medullae secrete epinephrine and norepinephrine in response to action potentials rather than circulating hormones. Such pathways are called neuroendocrine reflexes because they include both neural and endocrine components. We'll consider these reflex patterns in more detail as we examine specific endocrine tissues and organs later on. In the nervous system of 168, you should have noticed that the sensory receptors provide complex information by varying the frequency and pattern of action potentials in a sensory neuron. In a similar way, the endocrine system sends complex commands by changing the amount of hormones secreted and the pattern of hormones released. In a simple endocrine reflex, hormones are released continuously, but the rate of secretion rises and falls in response to humoral stimuli. For example, when blood glucose levels climb, the pancreas increases its secretion of insulin, a hormone that stimulates glucose uptake and utilization. As insulin levels rise, glucose levels decline, reducing the stimulation of the insulin secreting cells. As glucose levels return to normal, the rate of insulin secretion returns to resting levels. In this example, the responses of the target cell change over time because the effects of insulin is proportional to its concentration. However, the relationship between hormone concentration and target cell response is not always predictable. For instance, a hormone can have one effect at low concentration and more exaggerated effects or even different effects at high concentrations. Several hypothalamic and pituitary hormones are released in sudden bursts called pulses rather than continuously. When hormones arrive in pulses, target cells may vary their response with the frequency of the pulses. For example, the target cell response to one pulse every 3 hours can differ from the response when pulses arrive every 30 minutes. The most complicated hormonal instructions from the hypothalamus involve changes in the frequency of pulses and in the amount secreted in each pulse. So, as we move on to the next module, there are a couple things that I want you to consider and I want you all to keep in mind. So, one of those is how could you distinguish between a neural response and an endocrine response on the basis of response time and duration? 2, how would the presence of a substance that inhibits the enzyme adenylate cyclase affect the activity of a hormone that produces its cellular effects by way of the secondary messenger CAMP? And 3, what primary factor determines each cell's hormonal sensitivities?